Okay, I know we didn't cover all that on Wednesday, but are there any questions about Wednesday? No. Well, one of the more important things on Wednesday is to remind you again of the purine pyrimidine groups. Hopefully you don't take those wrong. The other important thing to remind you about from Wednesday is the gene expression, DNA to RNA to protein, transcription and translation coupled together. You take what is in our genotype and turn it into a phenotype. There's also an important thing to think about holistically for the entire class is we talk about phenotypes a lot. It's also important to remember genotypes play a very critical role in what the phenotype could actually be. Today we're going to look at some things that are going to affect our ability to translate proteins. And these are things that are going to help with sort of maintaining translation working properly. It's not really the best way to describe it exactly. But there are some things in play that sort of make translation a little bit less complicated and a little bit I'm curious what it was. I'm just curious what was it. Come on, whose one was it? That's up.
Mine.
What was it?
Demaglio.
What was that? Just something or
Yeah.
a name or?
Just something.
Okay, fair enough. But that's what I was talking about. There are some things that we need to consider that are going to help keep proteins keep the same amino acid sequence. But what's really important is when we change DNA through mutations, what effect does it have on our proteins? And will it have any effect? Now the first thing is not actually going to help us keep the protein the same amino acid sequence, but it does sort of simplify things. There are 61 sense codons. I don't know if you looked at the math from the previous slide. When you use groups of 3 and there's 4 letters, there are 64 possibilities of combinations. Which is why there's 64 codons on the genetic code. And 61 of those are the sense codons. And through sequencing the genomes of lots of different types of organisms, we have noticed a phenomenon that is called codon bias. In each particular organism, one codon shows up more often than another codon, at least for the same amino acid. So for the case of redundancy, there are multiple codons for most amino acids. It turns out that cells, one of them is used more often than another. Sometimes it's as much as 90 to 10%, sometimes it's 60-40, sometimes it's even sort of evenly split 50-50 between 2 different ones. But is this by design? Is it fate? Is there some sort of controlling thing that makes one codon more common than another codon? I mean, statistically speaking, why should one be more prevalent than the other? They should be equally distributed if everything is equal. The fact that they're not evenly distributed tells you that things are not equal. Related to this idea, from the sequencing the genomes of lots of different organisms, scientists also noted that cells do not contain the tRNAs or 61 codons. They only contain about 40 to 41 tRNA genes. So in the genome of an organism, they don't have the ability to deal with all 61 codons at all, just some of those codons. And that is sort of what helps explain codon bias is you can use one preferentially because that's the tRNA that goes with it. And you have that one, so that's the one that shows up more often. So with codon bias, this is the idea that we don't have a tRNA for all 61 codons, which will be an important sort of thing to consider when we go forward to something a little more in-depth to think about. But codon bias doesn't really have any impact on mutations at all. It's just we are more likely to find one codon for an amino acid rather than all codons for an amino acid. But it does change from organism to organism, which is also kind of weird, or maybe makes sense. With the genetic code, redundancy is a really important thing that helps maintain proper translation of the protein. And by that I mean we get the same amino acid sequence of the protein. And so because of redundancy, you can make some small changes or a few changes. And as long as the codon changes to the right codon, the amino acid doesn't change because of redundancy. And so if we make a change in that third position, we're going to see that sometimes we get the same amino acid, sometimes we don't. Which is why understanding purines and pyrimidines is important. Because what did we see with the genetic code? What is more likely to give us the exact same amino acid? A purine-purine switch or a pyrimidine-pyrimidine switch? It's either. Going from purine to purine or pyrimidine to pyrimidine almost always gives us the exact same amino acid. There are two exceptions, but for everything else, purine to purine, or I say it's pyrimidine to pyrimidine, purine to purine, same amino acid, purine, pyrimidine, all sort of the same. So when you go from purine to purine, you get the same amino acid. When you go from pyrimidine to pyrimidine, you get the same amino acid. So redundancy is important because some changes still give the same amino acid. And that's an important thing. The translation machinery is designed to sort of deal with a few types of mistakes. Now, one of the more interesting ones that we need to think about is on the next slide, because obviously I start talking about the slide before I actually show the slides that are in between them. But when we look at the nucleotides of the codons, and looking at this sort of switch between nucleotides and nucleotides, the first two nucleotides of a codon are typically the most important for defining what amino acid goes with that codon. The last one is considered a variable position, because we can change that third position and still get the same amino acid. It's not always going to be true, but changing the third nucleotide of a codon will more than likely give us the same amino acid if we go in a certain way. Purine to purine or pyrimidine to pyrimidine will typically give us the same amino acid at that location. If we go the other way, purine to pyrimidine, pyrimidine to purine, we may change the amino acid. So redundancy is not foolproof, but it does give us a little bit of leeway, a little slop in the translation machinery. Now, related back to the idea that cells don't have 61 tRNAs, is this idea that was proposed by Crick called the wobble hypothesis or wobble principle. How do you deal with codons for which you don't have a tRNA for? That's what wobble is designed to explain. What does it mean if something wobbles? Not in the scientific sense, just in a general sense. Unsteady. So imagine you had a chair that wobbled and it had 3 legs. What would make it wobble? Not somebody shaking it.
One leg is shorter than the other.
One leg is shorter than the other. Typically you get a wobble situation because you've got this little rock to it. And in the case of wobble, we see there's going to be a mismatch occurring, but we're still able to use the same tRNA to give the same amino acid, but for the wrong codon. But it's still the right codon. And that's a weird thing to think about. So to try to illustrate wobble, I have some little cartoons here to think about. So here we have 2 different examples. We've got codon number 1 and codon number 2, both are for the exact same amino acid. And the tRNA that has the anticodon that matches this one in all 3 locations is shown here. But if we need a tRNA with the anticodon to match this codon in all 3 positions, we would use a different tRNA. And cells don't have both of these. They'll have one or the other. So whichever one you have, it's going to work best with the one that goes with it. Okay, base pairing rules. We just have to think about base pairing rules. The wobble says we can allow a mismatch to occur between the anticodon and the codon, and still things will work out. As long as the codon is still for that same amino acid. And there are going to be some differences we have to worry about. But what does wobble really mean? One tRNA can be used to recognize multiple codons. That's what wobble actually says. A single tRNA can be used to recognize multiple codons for the same amino acid. And so when we look at sort of how things match together, in this codon and anticodon pair go together 100% because they all match A base pairs with U in all 3 locations. This represents a mismatch because U and G don't base pair together. This also represents a mismatch because U and C don't base pair together either. But one of these is allowable, the other is not allowable. And the one over there is allowable. The question I put forth to you is, can you think about what makes a G-U base pair okay, but a C-U base pair not? I think.
GU is still purine and pyrimidine, but CU is a pyrimidine and pyrimidine.
Yeah, so we're still maintaining a purine and pyrimidine base pair, even though it's not the right purine. Going from A to G is a purine to purine swap. But here we're looking at the anticodon changing, not the codon changing. So what we really need to think about for codon is a little bit more particular. To show you that, we're going to look at this thing on the board here in a second. And so wobble says one tRNA can be used to recognize multiple codons for the exact same amino acid. And just for simplicity, we'll say that our anticodon sequence is going to be AAA. I'm writing it backwards so our codon will be in the correct orientation of left to right. But what codon will base pair with this with a 100% match? What nucleotide base pairs with A?
TT.
Unfortunately, we are talking about codons, so we can't use T's because T's are in DNA and codons are in RNA. We use what instead? We use U. So A and U base pair happens when we talk about RNA to RNA, which is the case for a tRNA in the anticodon and a codon in the messenger RNA. That has to be RNA to RNA. And so this is a 100% match. So this is not a wobble situation. But if a mutation happens and that last U gets changed to a C, the anticodon AAA will still work for this codon. Even though there is a mismatch happening here, this is still an allowable mismatch. This is what we call the wobble situation, where the codon and the anticodon don't exactly match, but it still works. And it still works because it's still a purine pyrimidine pair there. And for whatever reason, the opposite does not happen. But this is what wobble says. One tRNA can be used to recognize multiple codons for the same amino acid. A lot of students get wobble and redundancy mixed up. They sound similar, but they're actually opposites. Redundancy is multiple codons for one amino acid. Wobble, one tRNA for multiple codons. So see the symmetry in those definitions. Lucky for you, in genetics, we're not going to worry about trying to pick and say what codon anticodon pairs go together. But there is an important idea here. As the purine-purine swap, pyrimidine-pyrimidine swap, or purine-pyrimidine pairing is still what we're looking at when we talk about things. So some changes are okay. Some changes are not okay. And while this will work, if we unfortunately make the other change, if we go to G instead, this would not work. So this does, but this would not, just because it doesn't have the correct purine pyrimidine in there. And so if we go back through the genetic code, we can kind of pick out how many codons or tRNAs do we actually need. It's something that people have already done. And when you sort of count up how many different things you need, you generally get to about 40, 41 tRNAs, which is why every cell only has that many. When you look at our gene, for example, you need one for this one for that one. So you need 2, and 4, and then 6, and then how many boxes are there? 1, 2, 3, 4, 5, 6, 7, 8. You count them up, you basically get the appropriate number. So we have all sorts of things that are trying to keep us from making mistakes. But when we make a purine to purine switch or a pyrimidine to pyrimidine switch, it's a certain type of mutation that we're going to talk quite a bit about. Sometimes mutations will change codons, and when we just change one nucleotide for another, that is an important thing that's going to happen as far as changing amino acids. But one of the other important things about going from an mRNA to a protein is the idea of the reading frame. Remember, for transcription, a promoter and a terminator tell us the starting point and an ending point. For translation, we need a start codon and a stop codon. But a start codon is any AUG. And so, which AUG is the right AUG is the question. And this is in DNA form, but if you look for any ATGs in this sequence, which one is the actual start if there's multiple ones? Like here's an ATG, here's an ATG. Which one is the actual start of the protein? You can't really guess, but there is information in there that's going to make it important. And so reading frame says, how do we group things together as groups of 3? Because a codon is a 3 nucleotide sequence. When you look at this DNA sequence and you start grouping them in groups of 3s, you get different groups of 3s, which means you get different amino acids depending on which starting position you choose. So we needed something that's going to help us pick the right reading frame, which is something we'll sort of think about here in a second. Now looking up nucleotide letters is okay, but sometimes it's nice to sort of see something in action. So, anybody pick out any phrases in any of these 3 reading frames? There's a few of them. If you look close enough, maybe you will notice each one has a different kind of message. Maybe it doesn't make any sense. And maybe you could argue the one that has the sort of most sort of grammatical sense is the right one, but maybe it's not. And so a start codon is important because that's the first amino acid of a protein. It's not the most important thing in telling us sort of where we start. So keep in mind that how we group things together in groups of 3s is going to impact what amino acids we get and where we get them. We don't need to specifically remember what the name of the location is for allowing for the initiation of translation. But there is a sequence in the RNA near the start codon that tells you this is where to start. So that relationship between this piece of RNA or sequence and the start codon allows us to find the right start codon, the right start codon, and the right reading frame. Molecular biology, you'll hear more about this, how it works, the translation, initiation, sort of steps. But for genetics, we don't really care about it. But reading frame is important, not because we care about where we start. We care about what happens if we add something in the middle or remove something from the middle of our gene sequence. That's where a particular type of mutation will start to show up. So basically everything in the transcript is needed for the translation process. Some of it's needed to tell us where to start, some of it tells us where to end, and the most important part is what amino acids do we give and where are they positioned. Hopefully we already sort of remember basically that replication can make mistakes. Even though we have proofreading activity and mismatch repair, mistakes get made from replication. They're very rare, but they will happen every so often. Our cells being exposed to radiation of certain chemicals can also cause mutations or increase the mutation rate. And sometimes we have DNA pieces that move around and jump around or recombine in different ways and cause problems. So there's all sorts of things that can go wrong. And we need to have a way to sort of fix these things if we want to stay working properly. Now, first thing is, let's talk about the different types of mutations, then we'll talk about the mutagens that cause the mutations, and then we'll talk about the things that fix those mutations. And then we can think about what is the impact of these mutations on certain cellular functions, namely cell division and how that relates to cancer. So you can have mutations that affect the gene sequence or the promoter region. Those are the two places where we're going to see things become a problem. And if the protein or if the mutation is in the gene, it's probably or could have some effect on how the protein functions. And as we're going to see in a little bit, that could be a good change, a bad change, or a neutral change. If the mutation is in the promoter, that might affect how much or not the gene is actually expressed. And if you don't make enough of a protein, that can be just as bad as not having it at all, or making too much of it, or it malfunctioning. So they all sort of relate together as far as things that go wrong. We have certain names for these, but they're not on the next slide, they're on the slide after the next slide. So we'll come back to this idea of what we call these things. But most of us are probably familiar with what the definition of the mutation is. We want to call it a heritable change. So something that can be passed down to your either offspring or daughter cells, which are two different things. So if mutations happen in germ cells, those are passed down to your children. If mutations happen in a somatic cell, the mutation only exists in you, but every other cell that gets made from that one cell is going to be the same mutant cell. So we have sort of different things to worry about, things that are transmitted sort of vertically from parent to offspring, and something that just propagates within a single individual. So meiosis, mutations, that's going to affect your children, but mitosis problems is going to affect only you. That's sort of a good thing. Some things are not heritable in the fact that they go to the next generation, but they're heritable in the fact that they go to the next series of cells. When we separate mitosis and meiosis as different cell division sort of outcomes at the end of the day. But again, mutation is a random event, chance encounter with a mutagen or 1 in 10 minus 10 chance of making a mistake through replication of DNA. And it's a random event, and that's something that's really important about mutations. Cells don't make the change because they're exposed to something. It's sort of the other way around. They make the change, then they're exposed to something, and then we'll see what happens. That's it as a mutagen, and the mutagen does cause a change. So that's the story. A lot of people have a misconception on mutations when it applies to something like antibiotic resistance. For the longest time, people thought that our use of antibiotics and our exposure of antibiotics to bacteria made them resistant. A lot of recent studies have shown that bacteria are already resistant. It's just that when we expose them to antibiotics, we eliminate the fact that they are already resistant. So they have these random mutations, some of them are helpful. Again, when we have a mutation in the gene, we typically affect the functionality gene, which could either be a loss of function or a gain of function. Gain of function means it could do the same function but better, or it could do a new function. And new functions is how we get evolution. I think back to the idea of paralogs, or is it orthologs, which is the one that happens in the same organism. What'd you say?
I don't know.
Okay, does anybody remember if it's ortho or para? Paralogs, typically things we find in duplicated gene in the same organism. And what happens to that paralog? Sometimes it gains mutations where it becomes non-functional. Did anybody remember what we called that thing that would result from mutations in activating a paralog? We called it something gene. A pseudogene, yes. And so loss of function is something that can be happening. But if that's paralog gains mutations that make it different, then we have evolution going on, where we have some new function or an altered function that changes things. Now, the other thing is nothing can happen. It's always, neutral is always a possibility. Nothing is changed. Same thing is true down here, neutral, even though it's not listed, no change is always a change or a result. There's a change, But is there actually an effect? Not always. So hypo and hyper, hopefully we're all very familiar with hyper and hypo, with things like hypo and hypertonic, hypothermia, hypertension, hypoglycemia. Do you know any other hyper and hyper words?
Hyperactive.
Can you be hypoactive? Have you heard anybody being hypoactive? It's not common, but yes, if you're like a sloth and in a coma, maybe you're hypoactive because you have less activity than someone normal. But hypomorphic and hypermorphic, a lot of people misunderstand these phrases because morph and morphology and shape go together. We're not changing the shape, we're changing the expression patterns. And it's because we're having a mutation in the promoter. But hypo and hypomorphic mutations are kind of boring. The mutations that affect the gene are where we can see different types of mutations that actually show up. That's where we're going to spend most of our time. Generally speaking, there are different classifications of mutations. First is what we call a substitution, where one nucleotide gets changed into a different nucleotide. The other is called a frame shift, where we add or remove a nucleotide which changes how the threes are grouped together now. And then we have something that's going to increase the number of repeat sequences, which could also decrease, so it could go up or it could go down. Does anybody remember the polymorphism that'd be most likely associated with a substitution? Does anybody remember the three different types of polymorphisms that we talked about for an earlier test? Was it test one? Yeah, it was test one. But we need to now dredge that up a little bit. Should I tell you what they were alphabetically? We had the C and V. We had the S and P. And we had the STR. Is this ringing any bells with anybody? Slight bells? We call this the copy number variations with really large changes, which is kind of more like the chromosomal deletions and duplication events. Single nucleotide polymorphisms, where we see substitutions come into play. And STRs, remember what STR refers to? Short tandem repeats. Remember, sometimes the repeats are long or short. Basically, STR is what we sequence when we try to identify somebody, if you're doing Ancestry.com or 23andMe. And so these are actually going to sort of change the repeats. They're going to be directly related to STRs. We're going to see as we go forward, you're going to be reminded of these things in some slides in a minute. But I want to see if you can remember before we sort of see them. So mutations are possible from replication, and we can make those mutations more likely to occur by exposing the cell to mutagens like radiation and different types of chemicals. But as you can see, one of the things that is different between your replication errors and your mutagen errors is we no longer see the repeat sequences changes, but we also see DNA breaks become a possibility. So we don't always see the same types of changes with the different types of sort of errors inputting units. And these we're not going to talk about so much anymore because we've already sort of dealt with these quite a bit. But can't forget that sometimes crossing over makes mistakes. These are large scale changes. But they can cause frame shifts. That's sort of one thing that they will sort of sometimes cause is frame shifts can occur. And so first we have the simplest case, which is the substitution. And again, in a substitution mutation, we change one nucleotide for another nucleotide. And we call the two different types of substitute mutations either a transition or a transversion based off of what nucleotide gets changed to what nucleotide. The effect of the mutation is either classified as silent, missense, or nonsense. And so this tells us how the DNA sequence changed. This tells us what is the impact on the protein sequence. So we want to separate those two things. How do we change the DNA? And how does that affect our protein at the end of the day? And transitions and transversions go directly back to understanding purine, pyrimidines, and redundancy, and wall, because they're all tied together with the same basic concept. Transition mutations are defined as purine to purine or pyrimidine to pyrimidine. These are the most common type of mutation that we see. They're about twice as likely to occur compared to a transversion. It's either because of a mutation, a mutagen affecting a change, or it's because of replication DNA polymerase adding the wrong nucleotide. It's really important to understand the transitions of purine to purine or pyrimidine to pyrimidine. And this falls directly in the wheelhouse of redundancy is going to be okay. We should be okay if we have a transition, at least in one place, but not the other two places. Transversions are almost always bad. That's where we go from a big nucleotide to a small nucleotide, or vice versa. And that's what makes it really bad, because we see the size difference go wrong. Our base pair is either too small or too big, but not the right size. And being the right size is important, even though it might not be a mismatch. Being the right size is still sort of okay. Which again, something we want to think about is changes that sort of mess up this purine and pyrimidine sort of pairing are less likely to occur because they're not allowed and they cause more problems. So I wouldn't say that there's a grand design of things, but at least is an evolutionary, what's the word, not selection, but sort of impetus, not the word either. I can't think of the word I want to use. Anybody have any ideas for what word I want to use? It's not a calling, it's not a desire, it's like a thing. I'll think about it in a second. First I have to stop thinking about it, and then I can remember what it was. So we're making a change. Substitution is all about changing one nucleotide. And that means a single base pair is going to be different. The silent mutation is called silent because no protein change is detected. We change the DNA, we change the RNA, but the same amino acid sequence is the result. And this is all due to redundancy. Without redundancy, silent mutations don't exist. Without wobble, these also don't exist as well. So that's why wobble and redundancy are really important in thinking about the link between mutations, between DNA and proteins, is to really allow silent mutations to exist. We can get some changes, but they don't really do anything. These silent mutations are most of the SNPs that we find in people's genomes. We get slight nucleotide variations that don't really cause a effect. It's a pretty important sort of thing. Missense mutations can be classified as SNPs, but SNPs are typically the things that don't make a difference. That's why they're sort of useful. But silent mutations, same amino acid sequence, because nothing changed. Missense means we get an amino acid, but we get the wrong amino acid. How bad is this going to be? This is something that is actually really hard to describe. We'll talk about more as we go forward. But in a missense mutation, only a single amino acid is changed in the protein sequence. Typically, proteins are hundreds, 500 amino acids in length. So usually, like 300 amino acids is the typical length of a protein. How important is that one out of 300? Really depends on a couple of factors that we'll talk about. A silent mutation is a neutral change. A missense mutation has different possibilities, good, bad, or neutral. Nonsense mutations are always going to be bad changes. They're going to have a negative effect on protein function. They're called nonsense because in this type of change, a stop codon is present early in the coding sequence of the RNA. So we can make one change, and that makes a stop codon in a place it's not supposed to be. And that creates what we call a protein truncation, a short protein. And here, sometimes length is sometimes important. If you get 99% of the protein, maybe it's okay. If you get 50% of the protein, probably not okay. you know, Halloween's over, but it's still the Day of the Dead. So imagine you're giving birth, and the doctor just goes like this with a samurai sword halfway through the birth and says, here's all you get. Are you gonna be happy or sad? Why, you got half a baby, is that good? Isn't half better than none? Yeah, in this case, it's, yeah, true. Half is probably worse than getting just, none. That's what sort of it is. You try to make something a certain length and you get like a little bit of it, you're gonna be pretty upset. And as I was saying, sign mutations are always neutral. No change in functionality, good, bad, or whatever. When a sign mutation occurs, we call this a synonymous change. Does anybody know what synonymous means? If you were to call something synonymous or two things synonymous, what do you typically say?
It's the same thing.
Yeah, they're the same, right? So, same amino acid. That's the whole idea of a sign mutation, is we get the same amino acid. DNA changed, codon changed, but still same amino acid. And transitions are more likely to occur than transversions. Silent mutations are more likely to occur than the other mutations. We have a lot of sort of things that try to contribute to keeping proteins being translated properly by sort of pigeonholing us into these are the changes that are okay, and other changes are going to be bad, and that typically means cells function worse so they don't reproduce and don't survive. But these are okay mutations, and we allow them to occur. And so synonymous changes more likely to occur than what we call non-synonymous changes, and transitions are more likely to occur than transversions. But if we dive into the molecular biology of why does this line mutation actually work, we need to think about our codon and which positions are variable and which positions are not variable. And remember, the third nucleotide of the codon is the one that can be changed without changing the amino acid, especially if it's a transition change. So again, down here we have serine versus arginine, but U to C, that's a transition, so we still get the same amino acid. A to G, also a transition, still the same amino acid. If we do a transversion, G to C or G to U, different amino acid. So that third nucleotide becomes a variable position, so we can change it and still get the same amino acid. But it does have limitations. It has to be a transition, or most of the time, say 98% of the time, a transition will still give us the exact same amino acid. The only exceptions are going to be right here with the amino acid tryptophan, which if we transition here, we get a stop codon, or when we have the methionine codon, transition here gives a different amino acid. So almost always, transition gives us a silent mutation or a synonymous change. We don't need to worry about the exceptions. Just the general rule is transition in position 3 of codon, okay. Transversion, half the time okay, half the time not okay. First nucleotide change, bad. Second nucleotide change, also bad. Except under rare conditions. Again, we don't care about rare conditions, just care, generally speaking, what's going to happen. So silent mutations are most often caused by change in the third nucleotide of the codon. Missense mutations and nonsense mutations are caused when we change either the first or the second nucleotide of the codon. These where they're more likely to occur to give a change. And again, missense mutations can be good, bad, or neutral depending on the type of amino acid change that happens and where in the protein the amino acid change occurs. We call missense non-synonymous mutations because they're a different amino acid. Does anybody got to take biochemistry? Does anybody care about biochemistry and enzyme functionality? Active sites and things like that? Do you remember the different types of forces that hold together a protein, like ionic bonding, disulfide bridges, and things like that? If you change a cation amino acid, a positively charged amino acid, to something else, how's that going to affect the protein? And then something that doesn't have a base pairing partner or an ionic bonding partner anymore. If you take a hydrofluoric amino acid and turn it into a charged amino acid, how does that affect the protein? If you change the protein or amino acid in the active site, maybe it doesn't bind substrate anymore. So you really have to know the protein to sort of predict what these things are going to do. So they are the most variable type of change. But again, when we see missed mutations, it is most often caused because of a change in the first or the second nucleotide of the codon. And it can be either a transversion or a transition happening at this location. Now, the other thing that is also going to help give us a missense is this transversion in the third position. So transition to the third, typically associated with silent. Any other change anywhere else, typically associated with missense. Or it could be nonsense. But nonsense does require a specific type of change. There's only 3 stock codons, so there's only certain things that you can do to make this work. To give you an example of a missense mutation, and you think about how do we evaluate mutations? We have the sickle cell anemia disease. Is anybody familiar with sickle cell anemia? It's a problem with your hemoglobin protein that doesn't have the same oxygen binding capability as a normal protein. So there's a single nucleotide change in the gene sequence of one of these subunits that causes a change from a charged amino acid to a non-polar amino acid. This changes the shape of the protein, which causes the red blood cells to also be a different shape. They have this basically elongated sort of sickle shape, which is why it's called sickle cell disease. Now, the ultimate impact or the immediate impact of this mutation is these proteins don't bind oxygen as well as normal proteins. So would you call that a bad change or a good change?
Bad.
And on its surface, the protein is not functioning as good, so that's a bad change. But it turns out that sickle cell anemia is also correlated with the resistance to malaria. So for whatever reason, people that have this disease are also less susceptible or have a protection from a disease that can actually kill you. So is that a good thing or a bad thing to be protected from a disease? So that's the trade-off. Yes, we have a protein malfunctioning, but we gain resistance to a deadly disease. So as we think about things, is something good or bad is kind of your perspective on things. And would you rather be suffering from sickle cell anemia, or would you rather be suffering from malaria? Malaria is more likely to kill you and definitely has a lot more severe symptoms, but you can be cured from malaria, you can't really be cured from sickle cell anemia. At least not yet. We see a lot of this where protein malfunctions, and that protein's not functioning well anymore, but the ultimate result is we get some kind of benefit because we're sort of losing something. I'm not going to ask you to remember this specific variable, so just trying to illustrate some things. And lastly, the last substitution mutation we have is called the nonsense mutation, which will always be a bad change because we get a protein that will be shorter than what it should be originally. And shorter proteins obviously are going to be not as good as longer ones. And just like with missense mutations, what's going to happen here is typically a transversion or a transition in that first or second nucleotide of the codon. Sometimes a transversion in that third nucleotide is also going to be a problem. And sometimes even transition to that third nucleotide can cause a stop codon to show up. But what I want you to really understand and sort of keep as we go forward is silent is tied to a transition at that third nucleotide of the codon. And all other mutations are tied to typically the first 2. Or a transversion, and that third one is also going to give us a change. Anybody have any general questions about a silent, missense, or nonsense mutation? Again, silent definitely refers to single nucleotide polymorphisms. Missense, probably, but nonsense, probably not. Typically, we want you still sort of be sort of normal. Typically, the whole idea of SNPs is they don't cause and effect, which is why we sort of look at them as markers. Next we have what's called the frame shift, and here we are adding one or more nucleotides or removing one or more nucleotides. This can actually not cause reading frame changes, even though it's called a frame shift. There's one particular instance where reading frame is still maintained, but a change is going to occur. Does anybody have any ideas on what type of change we could make that doesn't affect the reading frame? Has to do with the number and the amino acid and the codon.
Stop codons?
No. That's a possibility. A frame shift could make a stop codon, but that's not what we're talking about here. How can we still make the same protein even though we have a frame shift?
Redundancy.
Sorry, what?
Redundancy.
No, I'm looking up for a number to think about. Some kind of number. And the number is 3. What happens if we remove 3 nucleotides? How does that impact the reading frame? You could potentially just remove one amino acid if they're all next to each other. If they're sort of spread out, then that's eventually we get back the same reading frame. But changes of 1, 2, or 4 are definitely going to change the reading frame, but changes of 3 or multiples of 3 are not really changing the reading frame. So they're a different type of mutation. So when we talk about frame shifts, we're typically adding 1 or 2 or 4 or 5, but not multiples of 3. Because there's a different mutation that has a multiple of 3 that we need to think about. Substitutions can happen anywhere at any time, but frame shifts are more likely to occur in certain locations. And typically it's when there is a run of the same nucleotide. It doesn't have to be this many lengths, but any multiple of 3 or 4, sometimes it's easy to get lost in how many did I just do. So if you're just adding the same nucleotide over and over again, maybe you sort of skip one and so move on. And that's sort of what happens here. You're adding the extra one, you just sort of lose your place. But this is not going to happen everywhere because this doesn't show up a whole lot. We're having sort of the same nucleotide all over and again. Now to help illustrate the idea of how frame shifts are bad, I have my sentence here, which is stupid, I know, but I dare you and challenge you to make a better sentence only using 3-letter words. The best person will get a point. It can't be too long because it can't fit on the whole page. But the red cat ate the red rat. Red eats red. It's supposed to be fat cat. The fat cat ate the red rat. Then they have 4 different words, whatever. Here you can see an example of an insertion. So how does the message change? Quite a bit, right? It's gobbledygook. It's nonsense. It's not nonsense mutation. It's not actual words. And here we make a change. We still get another word. The red cat ate her. That's still okay as far as the sentence goes. It's not the same original sentence. So frame shifts are bad because when they occur, they cause changes after that point. Every amino acid after an insertion or deletion is going to be different than what it was originally. So we get a large number of amino acid changes with frame shifts. Substitutions only ever change one amino acid or make the proteins shorter. But frame shifts will almost always make something that is much different than what it's originally started as. And if you'll indulge me for a few minutes, there is an example of this in action. It's a weird example because it's not a single nucleotide in a frame shift, but it is a fairly large deletion that can cause frame shifts. But it turns out there's a particular protein that if you mutate, you can gain resistance to HIV and not get AIDS. But it's a fairly big mutation. It's a 32 base pair deletion, which is a pretty big frame shift. It's kind of interesting. On the surface, this protein is now not functioning as well, but it does give you an advantage versus other people in the society because you're not affected by a certain type of disease. So on Monday, we have one more mutation to talk about called the dynamic one. It's fun and changeable and different. And also is because of a different reason than all the other ones. They're sometimes fun.